We're going to explain how the polymerase chain reaction can be used to detect very small amounts of DNA. And I'm going to use the example of try a test to see if some cells are infected with a virus. Okay, so that might be blood cells taken from a patient who may have HIV infection, or it could be a swab from someone's nose who they think the patient may be infected with, say, coronavirus. It can work whether the virus is using a DNA as its genome or RNA. It works with both. Right, so let's see how the overall process. So what you need to do is you get a tube, and in the tube you're going to put a mixture of the sample, okay, from the person, and you're going to have to put a DNA polymerase in there called Taq polymerase. Sorry, Taq polymerase. Now, Taq polymerase comes from a bacteria which lives in hot springs. So that means that this polymerase, it won't be denatured like most enzymes over 37. It can take temperatures of up to about 70 or 80, and it doesn't get denatured. And also in there is going to be a mixture of nucleotides. They are the monomers from which DNA is made of. So they are nucleotides with the base A attached, so and with T, with G and C. So you're going to sugar, phosphate sugar base. So where a nucleotide is, and they're going to be polymerized by the Taq polymerase. Okay, so that's what goes in the tube there. Now, what happens is you have a sample of viral DNA, tiny bit of viral DNA, and that's double-stranded DNA there. Now, if you heat that up to about 70 degrees C, the hydrogen bonds here holding the two strands together will be broken, and you'll end up with two separate strands. And then you cool it down so the Taq polymerase can work, and the Taq polymerase will make a complementary strand against that one, a complementary strand against that one. And then you do another cycle of heating. That will separate these double-stranded DNAs out again. So you'll end up with that. And once again, you cool, allow the Taq polymerase to work. So it uses the nucleotide mixture to make a complementary strand there, and there, and there, and one there. So, and you can carry on with this heating cool cycle, usually about 60 times. It's usually automated. And if you do that, and you can see potentially you could end up with 2 to the power of 60 copies of your original one piece of DNA. So you end up with a very large amount of DNA from a tiny sample. Okay, then the DNA that you've made can be analyzed. Okay, well, there's a little bit more to that, so we'll talk about that now. Right, so this is supposed to be our bit of viral DNA here, and I've labeled here, this is the 3' end, and this is the 5' end. And just to remind you what that means is, here's a bit of DNA, and that carbon there, that's the 5, number 5. This is the ribose, so it's going to be 1, 2, and that one there is going to be 3. So there's 5, and there's 3, 5, and 3, and so on. So this one here, this would be called the 3' end of the DNA, and this up here would be the 5' end of the DNA. And if you remember, it's double-stranded. The other strand is going to run in the opposite direction. So the left-hand strand is going from 5' to 3. This other one will go from 3' to 5, the complementary strand there. So let's just remind you what that means. Okay. Now, Taq polymerase it only works in common with nearly all DNA polymerases, it only works in the 5 to 3' direction. And that's how it starts off at the 5' end. It moves down using this strand as a template, ending up going to the 3' end. Right, now what I've done here is, you need to know the sequence of the viral DNA. So this is you choose a sequence at both ends. So this is, I've only done, that is unique to the virus, okay? So this is a unique sequence here. You'll only find that in the virus, not in the human DNA. And this is another unique sequence at the other end. Now, mine, I've only drawn them 5 long because it's too laborious to write down 30 of them. But usually they're not 5 long. You wouldn't be able to find a sequence which is unique if it's only 5 long. They're usually about 30 long, 30 bases long. And also, you know the distance here. So let's imagine that distance from this end up to this end. You know how far this. You know that this is 1,000. Let's say it's 1,000 bases long there in this particular case. Okay, which would be a fairly typical way of doing it. Right, now in the mixture, in addition to the Taq polymerase, you need Taq. Right, you need Taq polymerase, and you need the nucleotides A, G, C, and T. You also need these things called primers. Okay, now what a primer is it's a short oligonucleotide. So an oligonucleotide is a tiny piece of DNA, perhaps like in this case about 30 long, and that has to be made, that has to be synthesized by chemical methods. So what you'll get here is this one is called the forward primer, okay? And it's complementary to the sequence at the end here. So the forward primer in that case would be T, C, G, A, T, and of course it would be about 30 long, not only 5 long. That's the forward primer. And at this end, we would have the backward primer, or the reverse primer. Now the reverse primer here doesn't have a complementary sequence, it has the exact sequence here. We'll see why that is in a minute. So on the reverse primer, you're going to have T, A, G, A, T, and that's the reverse primer. So these two oligonucleotides you'd have to have in your mixture of Taq polymerase and your nucleotides. And you'd want quite a lot of them. You don't want them to be short, you want them to be in excess. Okay, so what happens then is you cool the DNA down. This bit here. You cool the DNA down and you allow your primer, so this is the forward primer, you allow that to anneal to the complementary sequence of DNA. Now Taq polymerase can't work from scratch, it has to extend from something which is already there. So this will be the 5' end of the primer. This will be its 3' end. Taq polymerase comes in and it can fill in all of that. So it will extend that primer. It will keep synthesizing a bit of DNA, move all the way down. And it may go past this sequence, this TAG8, this sequence may go past there, that doesn't matter. It will do that. So what is the next step to do then is, once you've made that new piece of DNA, is to heat. And that, of course, will allow the two strands to separate. So I'll show you what happens with this. So the two strands have separated due to the breaking the hydrogen bonds and heating. Now, with your primers, you cool it down, you allow the primers to anneal. So what's going to happen here Well, if you think at this end, we will have made a complementary sequence to that, yeah? So at this end, we are going to have A, T, A, T, C, T, A, that end. And our reverse primer will hybridize to that perfect' So the reverse primer is going to go there. And then the Taq polymerase is going to, so also this is going to be the 5' end, yeah? The Taq polymerase is then going to extend this one, and it will keep going. And when it gets to here, it will stop, and it will stop there because there's no template for it. It also needs a template for it to work on, and it hasn't got a template, so it's going to stop there. So we've got our new bit of DNA there. Then once again, we're going to heat this up, break the hydrogen bonds between the two strands. So I'll do that. Move this down. Right, heat it up. Right, now allow it to cool, and the primers can anneal. Now where are the primers going to go? Right, we are going to get the red primer is going to bind to that. That's going to be complementary. I'll just draw it. I won't bother drawing the little bits on it. So you're going to get your red primer going there. And you're going to get your blue primer going here. That's the 5', remember? And this is the 5' here. Then the Taq polymerase is going to come along and it's going to fill in, moving 5' to 3 in that direction, making that yellow strand, and it will stop there. And again on this side, it's going to move 5' to 3. It's going to keep going and do that. Okay, so now we've made 4 copies of our original DNA, and you will have noticed that all of those copies are the same length as the distance between the 2' here, which I've done in purple. Okay, they're all going to be this 1,000 bases long. So you will have made, in that case, you will have made lots and lots of copies, well, only 4 of these fragments, and they're all going to be 1,000 bases long. You've done that 60 cooling heating cycles, you're going to end up with multiple copies of those. So hopefully you can see now why the primers are needed and why you need 1 at each end. And that the polymerase works on the 5' to 3' each time. You've got your multiple copies of that. Now how can you tell? So you've got a tube there, and it's going to have lots and lots of copies of the viral DNA. So here is our tube with the viral DNA in it. Now there are a couple of ways, there are several different ways in which you need to show that is the viral DNA. And one way of doing this is the simplest would be to show that all that DNA is 1,000 base pairs long. And you could do that by doing gel electrophoresis. So what you get a piece of jelly, agarose gel, very thin like that. It's going to have 2 little slots in it there. Okay, you're going to put your sample into one slot, and then you're going to put some standards into the other one. So I'll draw this now if you're looking down on it. So there's the one slot with the standards, there's one slot with our DNA in it, of what that you may be the viral DNA. Now electrophoresis, DNA is negatively charged, sorry. So you make that end there positive, 20 volts or whatever, and this end negative. And what happens is the DNA pieces are going to move that way. Because the smaller pieces, it's like a filter, the smaller pieces are going to move faster. So you're going to end up with a series of, in the standards, you're going to end up with a series of bands. I'll do them in blue, like this. These corresponds to different sized bits of DNA. So in the standard, you know that this one is going to be, let's say, that's going to be the biggest one, because it hasn't moved far, let's say that's going to be 2,000 bases long. Let's say this one is going to be 1,000. That one down there might be only 50 bases long, and so on. And then we're going to put our viral DNA in this one. So that goes up there. So you can visualize the DNA by adding a stain called histidine bromide and using UV light. And you can see that you've got this very dark band here at 1,000 bases long, which means we've made lots of bits of DNA the exact length that we expect. That's a strong suspicion that it is the DNA. So there was viral DNA in that sample. The other way of doing it is more specific. So you get another oligonucleotide called a probe. Okay, and you make this maybe 30 long again. I'll draw it less long, not as long as that. So T, T, A, G, C, C, T, A, whatever. And you know that this sequence is contained in the viral DNA. So if you mix this oligonucleotide probe up with the DNA sample, it will, maybe that's where it will anneal to say that bit of the DNA. It will hydrogen bond to it, the complementary sequence, and it will bind to it. And then you usually make this radioactive, or you can make it fluorescent. And then if you put it in there, you will find that it will only bind to the specific DNA sequence from the virus. So if you find you've got radioactivity incorporated into that DNA or fluorescence incorporated into that DNA, it means that you do have, so you must have that viral sequence. So that's a more specific way of doing it. Okay, now I've said the viral DNA all the way through that, but of course it will work on RNA, because as long as this first bit of DNA we had here, if that's RNA, it will still work fine. Of course, with RNA, you know, A's, they won't be A. They'll you don't have, sorry, T's you don't have in RNA. They'll be uracil. But apart from that, the primers can anneal to the RNA instead of the DNA. The DNA polymerase can work using that as a template, and you'll get the same sort of results. Okay, so let's leave that there.